So what do we study? Well, we study fear. Um, and well, what is a good definition of fear? So give me a second. I'll let you read the Yoda quote. Well, according to Yoda, fear is the path to the dark side. Fear leads to anger, and then anger leads to hate, and then hate leads to suffering. Very philosophical. But if you're to study a phenomenon such as fear, you really need to have a very good definition, right? You need an operational definition. And so the definition that I sort of, in some ways, inherited from my postdoc mentor's lab of fear, and I use the same definition to this point, to this date, which is the perception and recognition of danger, the learning and remembering of dangerous experiences, and therefore the coordination of defensive behaviors to environmental threats. So what I'm getting at here is a couple of things. One is our perception and recognition of danger actually requires largely the ability to recognize and learn what are things that are dangerous in our environment, right? And so when we experience something that's considered dangerous, we learn about those dangers. What things in our environment lead to those dangers, right? And what this allows us to do is coordinate appropriate behaviors to these threats that are in our environment. We call these defensive behaviors. Now, fear itself is normal, right? Without exhibiting fear, we probably wouldn't survive very long on this earth, right? Now, all of us probably think, well, when I'm afraid of something, how do I respond, right? Well, the reaction to potential threats can come in many different forms. The one thing that we might note is that our heart rate might increase, right? We also might show increases in sweating, right? Our pupils might dilate, as my daughter has pointed out. I don't know where she's learning this, but she's only 7 right now. Our blood pressure can change, right? Our breathing changes. We become more vigilant to the things that are around us, right? We might become more prone to startle, right? If all of a sudden you're exhibiting sort of certain, you're in a fear state, if somebody comes from behind you and taps you on the shoulder, you might jump, right? Others, you can exhibit a facial expression related to fear. We can also measure certain hormones, such as stress hormones. These become elevated. Now, one of the things that ties all of these things together is there are certain behaviors that are exhibited not just in people, but also in different other species of animals, which is when mammals are exposed to threats, they will show 3 types of behaviors generally. One is they will either freeze, remain motionless when they detect threat, depending on the severity of the threat or its location, right? They will remain motionless. So you might ask, well, why is that being motionless actually helpful? That doesn't mean that you might get hurt. Well, when we think about the natural relationship between a predator and prey, right? Say a lion, right? Hunting a gazelle or something to that effect, right? In some ways, freezing itself is adaptive because a predator actually responds to movement, right? And so remaining motionless, like, a squirrel, right, might freeze, for example, when you're walking your dog. It actually minimizes potential producing that chase response. Now, sometimes the most appropriate response is to run or flee, right? And animals will exhibit that depending on how proximal or how close the threat is. And sometimes the threat may be too close, and the only thing left to do is to fight. Okay. So these are, I think, commonly shared across different animals, not just in humans, or in rats and mice, which I'll be focusing mostly. In our laboratory, we're primarily focusing on using rats to study fear. Okay, so imagine here is a rat. I'm just going to give you an example potentially that might actually happen, even though this rat doesn't normally live in the wild. This is a domesticated rat here you're looking at. Let's say this rat is exploring this rock, and guess what happens? A snake pops up, right? And it attacks the rat. Well, the first thing that the rat will probably do is probably flee if it's being attacked, okay? And so it will either run or freeze. In this case, being attacked by a snake, right, already making this attack movement is to run or flee. Now, let's say that this rat decides it needs to get next to that rock for some reason. Maybe there is food that it hid somewhere, or there's a source of food, and it's willing to take the chance, right, to go past this rock again. Well, it might approach it by moving really slowly and then freezing, and then attending to sounds as well as visual aspects that might relate to the presence of the snake, right? So it's really important that the rat display a change in its behavior that allows it to tend to its environment and minimize movement and therefore attack. A snake again moves, will attack based on movement of a prey. Now, what I just try to point out here is a relationship that the rat here is learning about the relationship between that rock and that snake, because that snake perhaps resides under that rock. Whenever it was around that rock, that snake popped out, right? And so it's gaining a knowledge about the relationship. And so in some ways, this is a association, right? That rock, it becomes associated with the snake, right? And in doing so, that rat now has to display appropriate behavior under these conditions of threat. Okay. Now, this might sound very familiar to you if you think about Pavlovian conditioning. You guys have probably learned a little bit about Pavlov and classical conditioning. So my lab is interested in studying fear and the brain correlates of fear, okay, very broadly. All right. So how do we study this actually in the laboratory? I don't have snakes in my lab. I don't have rocks in my lab either. So what we do is our main subject, even though we use mice as well, are rats. And what we do is put them in what we call a Skinner box or an instrumental box that looks like this. Okay. And so the rat is put into the box and it's allowed to explore the box. Okay. So the first time the rat is in the box, it's learning about the environment in which it's in. Okay. And then all of a sudden, we give a mild foot shock. So see the grid floor that's in the image? We deliver a shock, so a little bit of current that causes a mild and momentary sense of pain in the paws, and that goes away really quickly. Okay. And so basically, the rat is learning that anytime this rat is in this box, that there's potentially something threatening in its environment, right? In this case, it's the environment of being in that box becoming associated with shock. Okay. Now, if we bring, so the idea here is that the context or this acts as the conditioned stimulus. Remember Pavlov, right? Where Pavlov's experiments were involved pairing the ringing of a bell with food. These experiments are pairing an environment, a box, with a foot shock, right? And in Pavlov's experiments, food, a bell, and food. This is our unconditioned stimulus. Okay. Now, if we bring our rat back, say 24 hours later, and we put him back in this box, and we're not going to shock him. We're not going to give a foot shock or anything. We're just going to put him in the box. And what this rat will do, upon being placed in it, it will start exhibiting a freezing response. Okay. It will lie still, even though this is not a video. The rat will sit there and display no movement at all. It'll go through periods of time. And so 90% of the time the rat is in this box, for example, or 80% of the time, it will freeze. It won't be exploring the box anymore. It had learned the association between the box and the shock, the foot shock that they got. And so in this case, the conditioned response is to freeze. Okay. Again, consistent with how how Pavlov described his experiments, we're describing it within that classical conditioning perspective. Okay. So here, fear is learned to the box, right? And how we measure fear is by measuring the freezing in these subjects. What percentage of the time do they freeze? That gives us an idea of how much fear there is. All right. Now, if that is how we study this, the behavior of the animals, right? These are the conditions that produce fear. The three main areas of the brain. So you're looking here as a drawing of the rat brain. And the three areas of the brain that I want you to focus on are, one, the amygdala. So the amygdala is part of the brain that's involved in emotion processing. So how we process emotion. And this has largely been demonstrated not just in people, but also rats and mice and in monkeys as well, that the amygdala is important for fear. Another brain region I want you to focus on here is the hippocampus. So the hippocampus is a part of the brain that processes the environment. It learns about where you are in a given environment in the world. So, for example, I'm in my office. My hippocampus knows about my office. I'm here every single day, maybe except Saturday. And so my hippocampus has a strong representation. It encodes information about its environment, right? Some days my office, I might be, it might be a tough day and I don't want to go to work. Maybe my amygdala is telling me that it's not a good day to go to work. All right. The third different brain region is the prefrontal cortex here in green. So the prefrontal cortex is important because it's generally described to be involved in decision making. And so it allows us, in some ways, it has a direct influence in controlling the amygdala, but also it's involved in communicating with the hippocampus. So the prefrontal cortex that sits, like its name suggests, in the frontal part of the cortex of the brain. So in the fear conditioning, the fear experiments that I described, The hippocampus is involved in coding the environment in which the rat is. The rat is learning about the environment, and this is being learned by the hippocampus. However, let's say that we introduce a foot shock. Well, that foot shock is going to cause the amygdala to become activated. Okay. Now, the other important thing to understand is that the amygdala and hippocampus are connected to each other. So the cells in the hippocampus, some of the cells can exit and reach the amygdala. And some of the cells in the amygdala, the neurons, they can send their long axons to form synapses onto neurons in the hippocampus. So they can communicate in both directions. Then we have the prefrontal cortex. The prefrontal cortex also can connect to the amygdala. Okay. And so information can go from the amygdala to the prefrontal cortex as well. So bidirectionality. Okay. And then the hippocampus and prefrontal cortex are also connected with each other as well. Okay. And so eventually, if the amygdala is involved in encoding, so let's start over here. So again, as I mentioned here, the hippocampus encodes information about the environment, right? And the shock is encoded by the amygdala. Now, the hippocampus also communicates to the amygdala. And the idea is that the relationship between this box, the environment, and the shock will become associated in the amygdala. And so just returning the animal back to the environment in which it was previously shocked will cause the hippocampus as well as the prefrontal cortex to activate the amygdala and then generate the freezing response in our subjects, or in humans, the sense of fear and dread. So, whoops, pardon me, I'm going to move some of these things around a little bit here. So my lab actually is focused on really three different aspects, three things about fear that are, I think, particularly interesting. One is, how does fear change depending on our developmental age, right? Everything that I've talked about is about fear in the adult animal. But as we all know that fear, we also can learn fear when we're younger as juveniles, just around that period right before we go through puberty, but also during infancy. Okay. And so one of the questions we're interested in is, how do the connections between the prefrontal cortex and the amygdala change across these different ages? And how does the hippocampus and amygdala connections change also? So that's one thing that we're interested in, sort of the development of fear over long periods of time. A second question we're interested in, and it's one that's also related to related to development, are sex differences in fear. Okay. And so we can study this both in male and female rats. And then we can look at how the amygdala here and how the hippocampus and the prefrontal cortex, which you don't see here, how are they activated? Are they activated the same way, males and females, right, by the same experience, or are they activated differently? Okay. Moreover, because when we say sex differences in fear, it doesn't mean just focusing on the adults. We can actually ask the same question in the juvenile animals. So that's the second part of what we do. And the last part of what we do is also, I think, particularly interesting. And I think the first two things that I described are about normal fear, right? Fear is normal. It's regulated by depending on how much threat there is in the environment, right? that we exhibit the right amount of fear so that we could survive. Now, what happens when the threat becomes very strong or traumatic? And so we're interested in how experiences that might be considered traumatic, how they impact later on fear. In fact, we're actually asking this question not 24 hours after a trauma, but almost 2 months later. So rats, for example, their lifespan, they live probably, no more than three years. And so their developmental time windows are very different. So we might ask, well, if the animal was exposed to a traumatic experience, let's say as a juvenile, what happens to their ability to learn about fear later on as an adult, months later, right? And so our lab is focused on studying this in different brain regions as well. What are the changes that happen, right? And so part of this is, of course, that this work has some relevance because it relates to or stress and trauma related disorders like post-traumatic stress disorder, which is a debilitating mental health problem in our country. And particularly, it's also more prevalent in females than males. So we're going to ask questions about the age in which trauma occurs and the sex of the animal. Does that matter in how it impacts, right, trauma and its effects later on in fear, right? So and I'm not going to get too much into the details here, is that one of the things that in order to establish how one area of the brain is connected to another, you have to go into the brain and inject these, what we call tracers that go into the brain. And so these are injected in specific parts of the brain, such as the amygdala. And these tracers will actually be taken up by the cells, and then they will travel to the origin of where the connection of another set of brain regions connects to that part of the brain. And so it allows us to look at the amygdala, but also label the brain, such as the prefrontal cortex here on the left, as well as look at how, so again, prefrontal cortex is connected to the amygdala, which is labeled here DLA, and then how the hippocampus is connected to the BLA, or the amygdala itself. Sorry for all these acronyms. There's a lot of terms. I don't want to go down that road right now. So this is work done by one of my first PhD students, Anthony Santarelli. And so he was interested in this question of looking at the connections between prefrontal cortex and amygdala at different age points. In infancy, so in a rat, infancy is between birth and about 19 days of age. In juvenile age, it's about 21 and just before puberty. And then adolescence is thought to be when puberty occurs. And then adulthood is about 75 days of age. So all of these regions make up the prefrontal cortex. So note here in the black line, this is the number of cells in the prefrontal cortex that project to the amygdala. What's particularly interesting is that the prefrontal cortex seems to be most connected to the amygdala during this juvenile period. And then it declines during adolescence and adulthood. There is one exception to this. It's a very specific portion of the prefrontal cortex that actually shows growth across these time windows. And if my daughter pops up behind me, because I had to pick her up, I apologize. And so she might pop up and say hello behind me. Anthony then looked at the connections between the hippocampus and the amygdala as well. So on the right panel here, are the number of neurons in the hippocampus that connect to the amygdala across these age points. So what you see here, unlike the prefrontal cortex, is that there's continued growth of connections through adulthood. Okay, so this is a very different pattern we see, right? Prefrontal cortex here in green is most strongly connected during this juvenile period. The connections are actually somewhat lost or pruned during adolescence, whereas for the hippocampus, growth happens. Okay, so that really sort of gets us at this point, suggests that, oh, these connections change their dynamic, they develop, and might suggest something unique about this juvenile period. Okay, now the one thing about this is that these experiments were all performed in male animals, male rats. So we didn't specifically study here in females. Now, part of that led us to the next series of experiments to examine sex differences and sex differences as they emerge from the juvenile to adult period. Okay. And what we're going to do is measure the activation of these three brain regions at three different in juvenile and adults, male and female animals. All right. So this work was performed by a graduate student, Loriana Colon. Excuse me, Dr. Loriana Colon. And so what she did here was to examine how context fear conditioning affects the activation of each of these brain regions in our rats. Okay. The first thing is that what you're looking at on the right here is the animal's behavior. So we're looking on this vertical axis here is the percent freezing, the percent time the animal is freezing to a box into the context in which it was previously shocked. We call this context fear retrieval because they're retrieving the fear of that context, the box. Notably here, what is really interesting is that in males, fear conditioning or freezing is rather low in juveniles and then increases in adulthood. In females, it's the opposite pattern. Freezing is highest in our female juvenile rats and then decreases during adulthood. So we see this really interesting pattern. Now, this is the, whoops, sorry, guys. The sex difference has been all before noted, but what we're arguing here, what we're demonstrating here is that this difference actually emerges uniquely, apologies again, and has a developmental pattern. Okay. So I'm not going to show you all of the data from this, but what we find is that if we measure the activation of the brain, the prefrontal cortex, we find that there's no difference in males and females in the activation of the brain when animals are exhibiting fear to the context. However, juveniles show greater amounts of activation than adults, irregardless of whether they're male or female rats. Remember I told you about the connections being peaking, the highest level of connections between prefrontal cortex happens during the juvenile period? Well, there you go. This is the activation of the brain is greater. The prefrontal cortex is greater than this time. The amygdala actually shows equal amounts of activation, whether the animal is juvenile or adult, or male or female. What about the hippocampus? Well, in juveniles, females show greater hippocampal activation than males. In adults, males show greater hippocampal activation than females. The pattern that we see with behavior corresponds very well with what's happening in the hippocampus. Okay. So the last set of experiments, one experiment I'm going to describe is looking at how a traumatic experience in these rats during the juvenile or adult period impacts the animal's ability to learn about fear later on. Okay. And so this is work by one of my graduate students right now, Samantha Hetherington. And so she's looking at juvenile and adult rats, female and male. And what she's doing is exposing these rats to either no stress, so one group has no stress, so it's a control group, right? One group has low stress, another has moderate, and another has high stress. And then 2 months later, they're brought into a novel box and they're just given a single shock. Okay. If we look at the females first, what we find here is that no matter whether it's low, moderate, or high, fear conditioning is enhanced 2 months later in the juveniles. Okay. In the adult animals that are given this stress condition, they're basically only group that showed higher levels of percent freezing or fear were the adults that were given high levels of stress. So low and moderate was not being low and moderate condition was not sufficient to produce any change. All right. Now in males, the pattern is almost completely opposite. If we're looking at males in the juvenile age, if they're given stress and we wait 2 months later, overall, we don't see an effect statistically of the stressor. So it doesn't produce a long-lasting effect. But if the animals were adults, the males were adults, and then we wait 2 months, we see what we get an effect where high produces the highest amount of fear learning, this point, and moderate shows lower, and then low has the lowest relative to the control, but they're all elevated. So the stress is actually enhancing fear learning, and it lasts for a couple of months. So this itself is pretty compelling, and we're looking at right now the activation of the prefrontal cortex. So we're studying a lot of really interesting things I don't have time to get into, but I think overall what our work has demonstrated so far is that the connections between within the regions of the brain that are involved in fear and decision making, as well as learning about the environment, continue to develop over the course, right, of infancy, during the juvenile period through adulthood. We're also pointing out here that there are sex differences. In fact, the sex differences seem to develop uniquely in males and females when it comes to fear and the activation of these same brain regions. And then ultimately, how is this clinically relevant? We think that if these are examples of normal fear and how that's processed by young and adults, male and females, these studies are getting at how fear can change depending on how much trauma you experience, right? Now, obviously, we're talking about rats, and so we can't really conclude too much about humans. But I think it starts beginning telling an important story about the timing of traumatic experiences, whether they occur in early life versus adult life, and how they might be uniquely affected. So I want to thank all of the students that have done the work. I'm actually missing a couple of students that have not been added here, graduate students and undergrads in my laboratory.